Welcome to this lecture on the comparative account of the vertebrate brain. Today, we are going to embark on a fascinating journey through evolutionary history. We will explore how the central nervous system adapted as animals transitioned from swimming in the oceans to walking on land and flying in the skies. By the end of this session, you will understand not just the anatomy, but the incredible story of survival written into the very structure of the brain. To begin our journey, think of the vertebrate brain as the central computer of a highly advanced biological robot. Why does this matter? Because the environment dictates the hardware. Just as a simple remote-controlled car only needs a basic circuit board to move forward and backward, a fish needs a relatively basic brain to swim and find food. But what happens when that robot needs to fly a drone or solve complex puzzles? You need a much more powerful and specialized processor. As vertebrates moved from water to land and eventually into the skies, they faced entirely new challenges, like breathing air, balancing on two legs, or navigating in 3 dimensions. To handle these new challenges, their central computers had to evolve, adding new parts and expanding old ones to ensure their survival. Now that we understand the brain as an evolving computer, let us look at its formal biological definition. In vertebrate anatomy, the brain is the highly specialized anterior enlargement of the central nervous system. But how does it form? Embryologically, it starts as a simple, hollow neural tube. Very quickly, this tube swells into 3 primary vesicles: the prosencephalon, or forebrain, the mesencephalon, or midbrain, and the rhombencephalon, or hindbrain. As the animal develops, these 3 regions further subdivide into 5 distinct regions: the telencephalon, diencephalon, mesencephalon, medencephalon, and myelencephalon. By comparing these specific regions across different vertebrate classes, scientists can trace the evolutionary history of the nervous system. We can see exactly which parts of the brain expanded or shrank depending on the specific survival needs of the animal. Building on this developmental foundation, we must ask why studying these differences is so important. Understanding the comparative anatomy of the brain is absolutely crucial because it provides a physical map of evolutionary history and behavioral adaptation. For example, by simply looking at the size of the olfactory lobes or the optic tectum in a fossilized skull cavity, paleontologists can deduce whether an extinct animal hunted by smell or by sight. In modern medicine, studying simpler vertebrate brains, like those of zebrafish or mice, helps researchers understand the fundamental wiring of our own human brains. Without this comparative framework, we could not test neurological drugs or understand human neurological diseases. Furthermore, it teaches us a profound biological principle. Form follows function, meaning the physical structure is perfectly sculpted by environmental demands. Knowing that brain regions change based on needs, how do we actually measure and compare intelligence across species? We cannot just look at absolute brain weight, because a larger animal will naturally have a larger brain. This brings us to a mathematical concept called the encephalization quotient. Conceptually, this formula compares the actual size of an animal's brain to the expected brain size for an animal of its mass. Let us break down the symbols. EQ represents the encephalization quotient, a dimensionless number. E actual stands for the actual measured mass of the animal's brain in grams. In the denominator, C is a constant that depends on the specific group of animals, like mammals or reptiles. M represents the total body mass, and R is the allometric scaling factor, typically around 0.75 for mammals. If actual brain mass increases while body mass stays the same, the EQ value increases, indicating higher cognitive capacity. Now that we have our formula, let us see this with a concrete example. Suppose we are studying a newly discovered species of mammal. We are given a total body mass of 10,000 grams, a scaling constant of 0.1, and an allometric exponent of 0.75. We are asked to calculate the expected brain mass for an average mammal of this exact body size. We start by evaluating the denominator of our formula, which represents the expected brain mass. We plug in our given values. The exponent 0.75 is mathematically equivalent to the fraction 3/4. This means we first take the fourth root of 10,000, which is 10. We then cube this intermediate result to get 1,000. Finally, we multiply 1,000 by our constant of 0.1. The final answer is exactly 100 grams. Physically, this means an average mammal of this size should have a 100-gram brain. Having established the baseline expected brain mass, let us take the next step and find the encephalization quotient. Let us assume biologists capture this mammal and measure its real brain mass, finding it to be 250 grams. We're asked to determine the EQ and interpret what it means. We use our EQ formula, which is the ratio of the actual brain mass to the expected brain mass. We place the actual measured mass of 250 grams in the numerator and the expected mass of 100 grams that we just calculated in the denominator. Dividing 250 by 100 gives us a final quotient of exactly 2.5. Physically and biologically, this final answer means the mammal has a brain 2.5 times larger than typical for its size. This strongly suggests the animal possesses advanced cognitive abilities and complex social behaviors. Now that we understand how to measure brain size mathematically, we must look at how the physical structure changes across different vertebrate classes, starting with fishes. Why start here? Because fishes represent the foundational aquatic environment. In fishes, the brain is primarily dominated by the midbrain and hindbrain. Because they live in a three-dimensional aquatic environment, they rely heavily on their optic lobes in the midbrain for vision. They also possess a well-developed cerebellum in the hindbrain, which is essential for coordinating complex swimming movements. Meanwhile, their forebrain is mostly dedicated to olfaction, or smell. This is exactly why predators like sharks have massive olfactory bulbs to detect blood in the water from great distances. Moving from the water to the land, we encounter the amphibians. Interestingly, when we look at amphibians, the brain actually appears somewhat simplified compared to fishes. Why would evolution simplify an organ? It all comes down to movement and lifestyle. Adult amphibians, like frogs, are generally sluggish and have much less complex movements than fast-swimming fishes. Because they do not need to coordinate rapid, continuous swimming, their cerebellum is drastically reduced in size. However, it is not all reduction. Their forebrain begins to show a slight enlargement. This crucial adaptation helps them process new sensory information as they transition to spending part of their lives on land. As we leave the water entirely and evolution progresses to reptiles, a major shift occurs in the forebrain. Reptiles face the harsh realities of a fully terrestrial life, requiring better survival strategies. To meet these demands, reptiles become the very first vertebrates to develop true cerebral hemispheres. More importantly, we see the very first appearance of the neopallium. The neopallium is a new layer of brain tissue specifically dedicated to higher order processing. This evolutionary upgrade is significant because it allows reptiles to exhibit much more complex survival behaviors, hunting strategies, and environmental adaptations than their amphibian ancestors. From the ground, we now take to the skies with birds. In birds, the brain undergoes another massive transformation, this time to accommodate the extreme physical and computational demands of flight. Flying requires constant, split-second adjustments. To handle this, a bird's cerebellum becomes enormous and highly folded, allowing it to process the rapid balance and coordination required to fly through the air. Additionally, their optic lobes are exceptionally large. This is because birds rely heavily on acute vision to spot prey from high altitudes and navigate at high speeds. The avian brain is a perfect example of how extreme environmental demands radically reshape neuroanatomy. Finally, we arrive at the mammals, where the evolutionary trajectory reaches a new level of cognitive complexity. In mammals, the forebrain takes complete control of the nervous system. The cerebrum expands so massively that it actually folds over and covers the midbrain entirely. To pack more brain cells into the limited space of the skull, this expanded outer layer, called the cerebral cortex, becomes highly convoluted with deep ridges and grooves. This incredible expansion of surface area is what grants mammals their unparalleled intelligence, advanced memory, and complex problem-solving abilities, allowing them to adapt to almost any environment on Earth. To truly appreciate these evolutionary shifts, let us look at this comparative diagram. Here, we see the brains of a fish, amphibian, reptile, bird, and mammal side by side. Notice the color coding. The forebrain is blue, the midbrain is green, and the hindbrain, or cerebellum, is red. Look at the fish on the far left. The green optic lobes and red cerebellum are prominent. Moving to the amphibian, see how the red cerebellum shrinks dramatically. In the reptile, the blue forebrain starts to widen. In the bird, the red cerebellum explodes in size and becomes folded for flight. Finally, look at the mammal on the right. The blue cerebrum has grown so massive and convoluted that it completely hides the green midbrain beneath it. This visual perfectly summarizes millions of years of adaptation. We have covered a tremendous amount of ground today, so let us review our key takeaways. First, we saw how the vertebrate brain evolved from a simple neural tube into a highly complex processing center. We learned how to use the encephalization quotient to mathematically measure relative intelligence by comparing actual and expected brain mass. We tracked the physical changes across species, noting how the cerebellum expands for complex movements like swimming and flying, and shrinks in sluggish amphibians. Finally, we witnessed the massive growth of the cerebrum in mammals, which drives advanced intelligence. Ultimately, the structure of the brain is a perfect physical reflection of an animal's ecological niche and behavioral complexity. You now have a solid understanding of comparative vertebrate neuroanatomy.